We will move on to our next speaker, Laura Niedernhofer. Are you there, Laura? And I love your background. It looks very interesting. So, Laura, whenever you're ready, you can just start.
Well, thank you very much, Morten, Daniela, and Alex, for the invitation to participate in this meeting. It's a little daunting following Jim. That was a tour de force. Jim, thank you very much. And I also wanted to say my thanks to the organizers for just maintaining the momentum in the field during these really challenging times. I think your work is critical, and I'm a huge fan, so thank you. So just following on Jim's talk, I want to talk about Senolytics briefly and their senescent cell targets. And like Morten, I really come at this from an angle of trying to understand the role of DNA damage in driving the aging process. And this started with work in Jan Huymaker's laboratory during my postdoctoral fellowship, where we began to characterize a number of DNA repair deficient mouse models and finding that they have many, many features of accelerated aging. So what I want to do is just share a couple of pieces of data on these mice where we've systematically compared them to natural aging. And I think this provides some pretty compelling evidence that lack of DNA repair really drives accelerated aging, and it doesn't just model a pediatric progeroid syndrome. So these are the ERCC1 deficient mouse. They're lacking a key enzyme necessary for multiple DNA repair pathways. And these animals reach adulthood looking quite healthy, but then decline very rapidly. And they spontaneously develop virtually every age-related disease that you can think of. So we think of this as quite systemic accelerated aging. And these mice have been therefore very valuable for screening senolytic drugs and other therapeutics that target fundamental biology of aging. So two key points about these animals were saying that they age rapidly because of the accumulation of DNA damage. So of course, it's very important to demonstrate this. And we did this in collaboration with Yinsheng Wang, who's at UC Riverside, and develops these very elegant assays that are very sensitive for detecting a spontaneous endogenous DNA lesions. And so these are cyclopurine addicts. They are repaired by nucleotide excision repair, which is an ERCC1 XPF dependent process. So we would expect these lesions, which are oxidative in nature, to accumulate in tissues of our ERCC1 deficient mice. So here's a histogram illustrating the cumulative total of these 4 cyclopurine addicts in renal tissues of our mutant animals in red compared to age-matched wild-type mice. And when the animals are quite healthy, they actually do not have increased DNA damage, despite their DNA repair deficiency. But by the time the animals look quite progeroid, they do have significantly elevated levels of oxidative DNA damage. But what I'd like to point out is this last bar over here is wild-type mice in old age. So it turns out that wild-type mice, even though they have DNA repair, do accumulate damage or have increased abundance of these lesions in old age. So this illustrates what's going on in the ERCC1 deficient mice is quite comparable to what happens with normal aging. And of course, DNA damage is one of the potent triggers of cellular senescence. And I'm not going to cover this at all because Jim did a wonderful job, but I just wanted to point to a collaborative review that we had on senescent cells and senolytics that came out recently with our colleagues at Mayo Clinic. But the point I want to make about this senescent cells is this was really an important moment to think about how DNA damage can drive aging, because not only do the damaged cells then become affected, but because of the secretory phenotype of senescent cells, they can also drive senescence and damage in a non-autonomous mechanism. So you don't have to accumulate DNA lesions in all cells to get this cohesive accelerated aging process. So we looked in our animals for markers of cellular senescence, and here are the ERCC1 deficient mice, again compared to normal aging. And across a variety of different tissues, we see varying levels of P16 messenger RNA. And I should point out, we really thoroughly characterize senescence endpoints and see very similar patterns, whether it's P21 or P16 messenger RNA, SA beta gal, or SAS transcripts or proteins. And what I'd like to point out here, though, is when you look at this heat map, you see that the pattern of P16 and other senescence markers is quite similar across tissues and across levels of these markers in the ERCC1 deficient mice. And this is at 4 to 5 months of age compared to animals that are 22 to 24 months of age. So again, it looks like an acceleration of a normal aging process driven by DNA damage. So we'd like to think of this as evidence that DNA damage does play a causal role in normal aging in mammals. So what we've been doing since then is trying to knock out ERCC1 XPF in a tissue-specific or cell type-specific manner to ask where is DNA damage most deleterious, or in other words, which senescent cell types might be most dangerous. And we found the answer in the hematopoietic system. So we knocked out ERCC1 using the VAV-I-CRE promoter, which hits all cells in the immune compartment, including stem cells. And we certainly see an attrition of cells. So here we're looking at peripheral leukopenia. Note that in young animals, the mutant ones are in red, we do not see a decrease in white blood cells, but there's a progressive decline as the animals age out. But we also see senescence. So when we look in splenocytes or bone marrow cells, we do see an increase in a number of markers of cellular senescence. Again, the mutant animals in red compared to naturally aged wild type mice in yellow. So the same thing happens with normal aging. And we see senescence increase. This is an example of looking at P16 messenger RNA in our mutant animals compared to H wild type and littermates of these mutant animals. So we see increased markers of senescence in T cells, B cells, NK cells, and monocytes and macrophages. So virtually every immune cell type. And we have a lot of deep characterization to still continue. But the point I want to make is we also found senescence across a number of solid organs. So this is an example of looking at P16 messenger RNA in bulk tissue. And even immune privileged tissues like the brain, we see increased senescence in these immune targeted ERCC1 deficient mice. And these markers of senescence are actually in parenchymal cells, so not in immune cells. So here's an example of looking at P16 messenger RNA, and the signal is co-localized with immunodetection of a protein that's specific to renal tubule epithelial cells. So what we're arguing is then senescence is being driven in trans or non-autonomously. And again, we have data looking at a number of different senescence markers. So we're accusing senescent immune cells of driving secondary senescence, but it's even more potent than that. It actually, the senescent immune system of these mice is sufficient to cause frank tissue damage. So here's an example where we see elevated liver enzymes in the serum of our animals. And this is the mutant animals again in red, H wild type in yellow. And so the liver is been damaged. We see proteinuria, so there's renal damage. There's serum amylase is elevated, so the pancreas is damaged. And when we measure muscle injury, it takes longer for that muscle to heal in our mutant animals. And in fact, the VAV-I-CRE mice are actually shorter lived in both median and maximum lifespan. And this is simply from a senescent aged immune system. So to test this more rigorously, that the damage is being caused by the senescent immune cells, we used adoptive transfer. So we take splenocytes either from our mutant animals or aged wild type mice and put them into a P16 reporter mouse. And then we measure total body luminescence over time. And in fact, when we transplant aged splenocytes, we do see an increase in senescent signal in these recipient mice. And please note that just aged wild type splenocytes are even more potent in inducing this signal. And we confirmed this by then measuring a bunch of senescence markers in solid organs of these mice. And in fact, indeed, senescent immune cells do drive secondary senescence in these recipients. So the kind of the dark and the bright side of all of this is an aged senescent immune system does drive senescence in trans, and it does so very potently. This is a cell non-autonomous mechanism, and it's sufficient to cause a lot of tissue damage. So I think this really gives us room to think about targeting senescent immune cells with Senolytics think they'd be an awesome target. And I think it's also points to the fact that immune cells might be really good biomarkers for biologic age, as has been shown by Ned Sharpless. So the other story I wanted to just amplify that Jim Kirkland introduced to all of you is we've been working very hard and closely with our Mayo colleagues to ask the question, could senescent cells be the cause of adverse outcomes in COVID-19? And that's because we noticed that diseases or being elderly, things that put you at risk of severe illness from COVID-19, all share in common a high abundance of senescent cells. So the unique paradigm that we had to test this hypothesis was actually quite simple. So this was an exposure paradigm created by our colleagues at the Center for Immunology at University of Minnesota, where you take your experimental mice and you simply expose them to pet store animals. So these are healthy young mice, but they carry environmental microbes. And to our horror, we learned very quickly that while young SPF mice can tolerate this, aged wild type mice cannot. And this is highly reproducible, that the old mice are dead within 2 weeks. And it turned out by serendipity that the pathogen that's killing these animals is a beta coronavirus that is closely related to SARS-CoV-2. In this case, it's mouse hepatitis virus. And we know this is the one that's killing the animals because when we look at day 11, when most of the old mice are dead, while pet store mice are carrying a number of different microbes, some of which are pathogenic, when we look at serology, the only evidence of transmission at this point is MHV. And when we look in tissues of these mice, we see pathognomonic lesions for MHV. So these are big syncytia that stain positively for MHV coproteins. And note that these lesions are apparent in the old mice, but not the young who have cleared this infection. Sort of the strongest evidence that it's MHV or this beta coronavirus killing old mice is when we gave them older young mice a sublethal dose of this virus prior to exposure to pet store animals, we could rescue them. So they develop an immunity against MHV. And here's the mice that have been inoculated, and old animals can now survive once they've been immunized against MHV, as opposed to the old mice that remain naive. So this gave us a paradigm in which to start testing senolytics. And we started with fisetin because it's a natural product that we view it optimistically might be very safe. And the paradigm we used is we used young or old mice and we exposed them to the pathogens. Then we started treatment, trying to get a translational component to this. And what we found is this treatment with senolytics was sufficient to suppress mortality by about 50%. And this is fairly reproducible. Similar results were obtained with dasatinib plus quercetin as a senolytic cocktail, as well as using the elegant Mayo models to genetically ablate senescent cells prior to NME exposure. So the mechanism of action we think is behind this is that senescent cells will cause an overreaction to pathogen-associated molecular patterns. So this is an example of data from Yi Zhu in vitro, where she exposed senescent or non-senescent cells to the classic PAMP LPS. And you see that the senescent cells, this SAS factor in the conditioned medium, it just shoots up if the cells are senescent when treated with LPS compared to non-senescent. The same thing happens in vivo. So if we treat old animals with LPS, there's an incredible amplification of these the SAS factors, causing a very pro-inflammatory environment. This was also true in the NME paradigm. So old mice that were exposed to MHV, they had an incredible increase in circulating levels of a number of cytokines compared to the young animals. And then when you treated the old animals with fisetin, we could see a decrease in these SAS factors in circulation. So we think this is really suppressing this hyper response to PAMPs that can drive cytokine storm and multi-organ failure. So another advantage with the senolytic treatment was we saw a decrease in the viral load, just looking at transcripts of MHV specific genes. And this afforded old animals more time to develop a proper adaptive immune response to this pathogen. So here's young mice, old mice that did not get treated and died shortly thereafter. And then this is from old mice that have been treated with Fisetin. They survive longer and they have an improved antibody response to this virus. So just in conclusion, this really spooks me because it means these old SPF mice that we're using to understand their immune system in response to infection and even vaccine, they really don't mimic what's going on in reality and may not be relevant to understanding what's going on in the elderly immune system. But Senolytics did reduce adverse outcomes in old organisms upon infection with a novel pathogen. And we think this really provides strong evidence for the geroscience hypothesis. I would have believed senescent cells contributed to a number of chronic conditions that are associated with old age, but to really protect against viral infection and maybe even be a target when thinking about immune responses to vaccine, I think this really stands up, really bolsters the geroscience hypothesis that the fundamental biology of aging is really affecting health in old age. So with that, I'm going to conclude by thanking my laboratory. These are all wonderful postdocs in my lab. A shout out to Matt Yusefzadeh, who led both of these stories. Most of the work is actually done by our amazing animal technologists that do a lot of the in vivo work. And we've got some great graduate students and undergraduates who are training and hopefully will stick with biology of aging. And I'd also like to thank my collaborators at the Center for Immunology and Mayo Clinic for their contribution to these stories. It's been a great collaboration. So with that, I'll stop and take questions if there's time, or let you get to sunshine and dinner.
Thank you so much, Laura. Really, really nice, amazing presentation. We have a question right down here in the front, and then Rudy after.
Does Fisetin kill both HUVECs and pre-adipocytes?
Yes, we do see some clearance of both cell types with Fisetin, but it's not going to hit all senescent cell types. So there's others where it's not able to clear. We need to test it on immune cells. I think that's where we're really lacking data.
That's great. Thanks, Laura. We have a question from Rudy.
Laura, thank you very much. Going back to your coronavirus and the old mice that you rescued with the Senolytics, just to give to have an impression about, let's say, the central role of the inflammation. Have you also tried to, let's say, rescue the old mice with anti-inflammatory drugs, whether it's in the IL-6 signaling pathway or dexamethasone or something like that?
We have not tried that in this particular exposure paradigm. I think it should be done. I think that's a great idea. My hunch is that would not be adequate unless you really bolstered the dose or had some sort of combination. But we were really focused on just trying to improve mortality at this point. The experiments are hard to do because so few animals live. And so we haven't gotten to that point yet, but it's a great question. Thank you.
Very nice. I would also like to ask a question. So the DNA damage that accumulates in the ERCC1 mice, what do you think that damage is? And is there a difference in the wild type, or what's the damage that accumulates in the wild type with age? What would you say?
I don't think we have the tools to answer that completely. It's just these assays are focused on a single addict at a time, and there's hundreds of oxidative DNA lesions. The pattern is slightly different in wild type mice, but it's clear that some nucleotide excision repair dependent products do accumulate with normal aging. The other thing is a lot of these lesions do kill cells, and then you can't detect them, and so it's really tricky. I think the important point I want to make is I shouldn't throw around the word accumulation either. I think there's actually a bolus of increased reactive oxygen species towards the end of life that really comes from mitochondrial defects and all these pillars of aging really being connected. And so I think that's kind of the same whether you're DNA repair deficient or not. It's just all of a sudden at the end, you just can't cope anymore with all of the different types of stressors.
It's a very, very good point. Thank you so much, Laura. I think we'll stop here. It was great to see you, and thank you so much for your talk. And